Hello, everyone. Welcome to this session for Oxford Nanopore Technologies. My name is Anna Disco, and I'm Associate Director for Business Development. We've got an exciting session for you prepared today with three amazing speakers. Before we start, maybe I ask who is completely new to Oxford Nanopore Technologies and doesn't really know what it is about? If you could put your hands up. Well, actually, it looks like most of you already know how it works, so that's great. Just to bring everyone on the same page, I'm just going to very briefly remind everyone what our technology is about. So we are obviously sequencing technology. We sequence DNA and RNA strands. At the heart of our technology lies a nanopore that's embedded in synthetic membrane. We've got motor enzymes that are attached throughout the library prep to the DNA and RNA strands, and they bring those DNA and molecule strands to the pore. We apply electrical current on that membrane, and as the DNA passes through the pore, the current is changing, which you can see in the form of that squiggle. Then we apply very elaborated algorithms that are able to deconvolute that complicated signal into actual bases. So what are the key benefits of our technology? We sequence DNA and RNA directly. That means that you don't have to use PCR, so there is no amplification bias. That means also that beyond the canonical bases, you are able to see epigenomic modifications. We are read length agnostic, so you can sequence very long strands, but also a very short one. So any applications that you are interested in, it will be appropriate. And we also are able to perform real-time analysis. We do provide a full end-to-end workflows, starting from library preparation recommendations through the sequencing and analysis as well. And if you would like to talk about any specific applications that you're interested in, please come to our booth, number 3053. And we've got a bunch of experts that can talk to you about anything that you are interested in. The one thing to remember about our technology is that we have exactly the same chemistry, exactly the same core technology in all our platforms. And it just basically, depending on the scale and the application that you are interested in, we would just recommend different platform. But basically, it's all the same, whichever platform you choose, which makes it easier to design experiments and then scale up, perhaps. But how does our technology apply to cancer and cancer research? So we all know that it's an extremely complicated disease, that it's now being really characterized on the molecular level. There's been a number of different mechanisms already identified that drive tumorigenesis. And we know that understanding all those different mechanisms is really important to a number of different applications in cancer research, like potentially, for example, finding new therapy targets, understanding treatment response for example, patient stratifications. But we also all know that we really need to be looking for those new biomarkers that will be driving that discovery further. So very briefly, I think we all now can acknowledge that so far cancer research was really focusing on understanding the impact of small variants like SNVs and indels on cancer. But I think we all know now that we really have to be starting looking more broadly and trying to understand all those different genomic and epigenomic variants on cancer mechanisms. That includes structure variants, being able to look at epigenetics, more precisely characterize gene fusions, but also look at chromatin conformations, but also be able, for example, phase those variants as well. All of this can really enrich your discovery. So with this, I will be moving on to our speakers, but I think, just wanted to summarize that I think really what we're trying to say is that precision oncology starts with that complete picture, being able to see all the different genomic and epigenomic variants, being able to haplotype resolve them, and hopefully that will enable the research to make further discoveries. Before we move on to speakers, just a very quick reminder, we've got a number of very exciting sessions happening at our booth. We've got live demos on two separate topics that actually will be partially touched on through the presentation of our speakers. We've got a live demos talking about our unique adaptive sampling enrichment method that doesn't require PCR. And we also have live demos that talk more about tumor normal end-to-end workflow as well. So please join us. You've got timings, but you can also find it online too. We also run data for breakfast sessions every morning at 9:30 that talk a little bit more in detail about all the different bioanalytical tools and different workflows that could help your research too. With this, I'd like to move to our first group of speakers, Mikhail and Aisha. And they will talk about the new developed tool, Severus, for accurate detection and characterization of somatic structure variation in tumor genomes using long reads. So Mikhail Kolmogorov is a principal investigator at NCI. Before joining the Cancer Data Science Laboratory in January 22, Mikhail was a postdoctoral fellow at University of California, Santa Cruz, supervised by Benedict Patent. Prior to that, he was postdoctoral fellow at the UC San Diego, co-supervised by Rob Knight and Pavel Pevznev. Mikhail completed his PhD in September 2019 in computer science from UC San Diego under mentorship of Pavel Pevznev. He will co-share this session with his colleague Aisha Keskus. Aisha has received her PhD from Bilkent University in Turkey. She is a postdoctoral research fellow at Mikhail's lab at NCI, and her research interest includes the characterization of complex structure variations in cancer genomes using long reads. Over to you guys. Thank you.
Thanks a lot for the introduction, and thanks a lot for having us here. Very exciting to present our work. I will make a brief introduction and kind of outline the research directions, and then Aisha will dive into more technical details about what we're doing. So we will be talking about structure variants, and many of you, of course, already know the importance to cancer. And but just to make sure everyone is on the same page, we're talking about any genomic alterations that are longer than 50 base pairs. And as you can imagine, they could be pretty long. They can affect one or multiple genes, sometimes called chromosomes, and can really deregulate cancer. And in particular, they are very heterogeneous, both in size and in terms of the patterns. And it makes it difficult to study them and difficult to draw associations as compared to single nucleotide polymer variants. On the pan-cancer level, we know that about 50% of the driver events are associated with structure, overlap with structure variants, so that indeed makes it an important mechanism in cancerogenesis. Typically, on a population scale, when people talk about population scale structure variants, they talk about insertions and deletions and sometimes inversions. In cancer, as you may know, they often have much more complicated somatic structure variants, such as chromoplexy, when you have a balanced DNA exchange between multiple chromosomes, or chromotripsis, when a chromosome arm or whole chromosome is shattered and then reassembled. And what we think is long-read sequencing technologies can help us to uncover the mechanisms and better characterize these complex structural variants. And in general, it's a very challenging problem because it's it can lead to chromosome scale copy number aberrations and complex rearrangements, as you can see on this example. And oftentimes, it is difficult to characterize it with short read because you need to deal with the cryptic signatures. So you have your coverage, unusual coverage profiles, and discordant read pairs. And then from this, you need to figure out what really happened based on the reference alignment. And with long reads, the advantage is if you're lucky enough and you read span the entire structural variants, then you basically have a sequence resolve view of your tumor genome. And you can also phase long reads, and that meaning that you can actually look at particular haplotypes. And so naturally, we wanted to explore different cancer types that are affected by structural variants. And osteosarcoma is one of these cancers. It's often driven by complex structure variants. It is a pediatric, most commonly a pediatric cancer. And it's relatively rare, so about 800 cases per year in the US. There have been really few new prognostic biomarkers and improvement in treatment and outcomes in the past 30 years, so we really need some new ideas how to improve this. At NCI, we have a comparative osteosarcoma program. We're basically studying osteosarcoma in dogs. In dogs, it's a much more common disease, about 10,000 cases per year. And it, of course, makes it an attractive model to study this disease. And so this osteosarcoma program is hosted at NCI, and Emily Blank is directing it. It's the same disease, but there are important differences as well. In dogs, it's unfortunately the long-term survival is lower. But on the other hand, the treatment is pretty different. We have access to large volumes of untreated tumor and adjusted normal tissues. And this is before therapy. So we actually are able to study the evolution of this, natural evolution of these tumors. So what we did, we took 2 dog models that were available and performed multi-site sequencing. So in each dog, we have multiple sites and also a matching normal sample. And each site was sequenced with a single flow cell, single R10 flow cell. Each yield about 30x depth, assuming the normal karyotype. So this is sheer DNA. We got really nice data. We were pretty happy with it. It worked really well. And this is tumor DNA extraction, so we didn't have any issues with that. And these are the coverage profiles for different sites. As you can see, there's quite a bit of structural variant aberrations. And so these are the 2 dogs, Oliver and Piper. And our goal was to really not just to look at the structural variants, but construct a comprehensive view of all genomic and aberrations and methylation, epigenetic aberrations as well. And of course, firstly, we wanted to focus on structural variants. And what we realized that most of the current long-read structural variant algorithms were really not designed for cancer genomes. They were designed using a quote-unquote healthy genome in mind and typically aiming for indels. So we decided to develop a new algorithm called Severus. And now over to Aisha, who will tell us about how Severus works.
Hello all. I'll briefly talk about the tool that we developed, Severus, which is a structural variation calling tool for long-read sequencing. The key point of my talk is going to be Severus is the best tool that you can find out there for the SV calling using long-read sequencing for the cancer genome. Why? Because we developed Severus with cancer data and for cancer data. So we use the cancer data to develop Severus. But Severus does not only work with tumor normal paired data, but also tumor-only samples and multi-sample tumor, just like Misha described for the canine osteosarcoma study. And then we start thinking about, we have this, very hard, challenging cancer genome, and we have the long-read sequencing with new information that we get, and how we can use this new information to solve this challenging cancer genome. So the first things that we get is, with long-read sequencing, we get improved phasing. That means we can directly assign each read as paternal or maternal haplotype directly from reads. So with a cancer genome context that we know to be highly heterogeneous and also highly consists of high aneuploidy, not using this phasing information would be a huge waste. So we incorporated that phasing information in our calls, and we provide haplotype resolved structural variation calls. And the other things that we know about cancer genome is it is unstable and evolving. So most of the events that we observe in cancer genome are actually a complex event that cannot be defined by a single line in a VCF. So in the last step, we cluster structural variations and annotate them. But to start with, building all these steps, we need to be sure that we are calling structural variations accurately. But unfortunately, there are not many data available to test if our tool is working well or not. So we sequenced our own cell lines. So we sequenced 4 breast cancer and 2 lung cancer cell lines and their corresponding normals with Illumina, PacBio, and ONT sequencing. Then we run 3 Illumina sequencing tools, MANTA, SVABA, and GRIDS, and 4 long-read callers, Severus, Savannah, Nanomon SV, and also Cinefus2, which is widely used for long-read sequencing. But for those cell lines, we don't have a ground truth, so we come up with these confidence call sets in which true variants are defined by supported by 2 technologies, at least 2 technologies, and 4 different tools out of 11. Then we using MANTA, we generated this confident call set, and also we calculate the F1 scores against this confident call set for each VCF. So here's our results for ACC 1395. In here, each color represents a different technology, and each bar is a different tool. You can see Severus is outperforming in both HiFi and ONT sequencing, consistently also in other cell lines. So at this point, we start, okay, we have this, nice data. But some SVs are particularly challenging for structural variation calling, and we next look at those, challenging parts. I'll briefly just talk about a few parts. So it looks complicated, but here we also have this, different technologies represented by different colors. We know insertions and tandem repeats are hard for Illumina sequencing, and we confirmed it our set. But what we didn't expect to see here is this breakend homology. And in these ones, we have these 2 breakends, one in the line 1 element, let's say, and the other one is in the other line 1 elements in a different position. And in those cases, we have an improved resolution with long-read sequencing. So with that, I'll just skip that part, but okay, sorry. So, it's not? Okay. So the other thing that I want you to focus on, this very last part, which call is not challenging, that almost all tools had a very high F1 scores. In those means, these are the rest of the SVs that we are just, finding not challenging. But except Cinefus2 in here. So Cinefus2 is designed for germline SV calling and doing quite well for genome, germline SV parts. But as I said in the beginning, cancer genome is hard, and you need to design your tool just for cancer. And all the tools here, except Cinefus2, is designed for cancer genome and doing pretty well job. So now we can detect structural variations quite well, and Severus is performing with, in all these challenging parts quite nicely. Next part, okay, we can start with building up in our, later steps. The first thing is improved phasing. For that one, our pipeline starts with phasing normal band file with germline variants. Here you can see we have haplotype 1 as yellow and haplotype 2 as purple. And you can also see, I don't know how clear it is, but phased SNVs. With using that phased SNVs, colored lines, we can also phase tumor sample. With that pipeline, we can make sure that normal haplotype 1 and tumor haplotype 1 refers to the same maternal or paternal haplotype. And you can also see the somatic SVs only occurring in tumor haplotype 2 in here. But you may ask why it's important in clinical use. So this is an example from our clinical collaborators. So let's say we have these 2 deletions. One is in exon 1 and the other one is in exon 2 in a gene mark, a very important marker gene X. When you just look at this one, you cannot say if these 2 deletions are on the same haplotype or not. Because if they're on the same haplotype, you can't have a wild type allele. you can just phasing, and you can just see, okay, that's the case. Both deletions are on the exon in the same haplotype, and the haplotype is wild type. So we might have a functional protein for this gene. The additional layer of information that we can get from here is all the reads in haplotype 1 is supporting this deletion in here. That means it's probably a clonal event. That means all the cells in your sample has this deletion. In other words, if we had like a multiple reads spanning through in haplotype 1, you could just say, okay, some portion of the cells has this deletion, some hasn't. So with Severus, we provide both haplotype information, also haplotype-specific varietal frequency that you can refer to clonality. Now we can, okay, so let me just skip this. Now we can identify the structural variations accurately, and also we can phase them. But the cancer genome analysis does not stop here because it's highly complex, and we need to get through these complex events. I'll start with this one example. Let's say you have this nice VCF from your structural variation calling, and you find this one structural variations going from chromosome A to chromosome B. The logical outcome would be it's an unbalanced translocation between chromosome A and B, leading to one rearranged chromosome. But let's say you have these two different SVs in your list, which is chromosome B to C and A to C. Now you have a completely different picture. Initially, we thought this can be like translocation, but now we can say it can be a template insertion cycle starting from chromosome A, goes to chromosome B, jumps to chromosome C, and then comes back to chromosome A, and then continues again, leading to a single rearranged chromosome. Or it can be a chromoplexy, just a synchronized translocation event between multiple chromosomes. In here, chromosome A, B, B to C, and C to A. Or it can be a reciprocal translocation event with an insertion from chromosome B. With complex SV clustering with Severus, we not only identify junctions that are in the same, in a single event, we also give you which of these three events is much more likely to be happening in your sample. So this is an example from our cell lines. So this is the interactive Severus graph that we output. We have these green rectangles. They represent the genomic segments. And these lines are genomic junctions. That means it jumps from chromosome 1 to chromosome 22. In this example, we have an insertion from 31 kb insertion of chromosome 22 inside the chromosome 1 in an inverted orientation. Here's another example. In this one, we have a fold-back inversion in chromosome 14. It just comes and then folds back. And then we have a small insertion from chromosome 5, and then it connects to chromosome X. It's even more complex event, a chromoplexy in ACC 1395. And in this case, we have 5 chromosomes involved, leading to 4 different derivative chromosomes. It can be even more complex in cancer genome. A chromotripsis in HCC 1954, although it looks like compact in here, the actual structure is something like this. So we can, all these examples that I showed you until now, they were all from cell lines which are known to be much more cleaner. But how it works with real-life tumor data, which is heterogeneous. Let's get back to the canine osteosarcoma study. Just to remind you that we have like 2 canine data, multi-sample in here. We run Severus for each canine separately in multi-sample mode. Here is the largest cluster in Oliver. We have a cluster templated insertions and duplications in here. So the next step, we check for, okay, how the copy number changing for this region. We run Vahan, a copy number, haplotype-aware copy number caller for long-read sequencing. In here, red represents haplotype 1, and blue is the haplotype 2. And if we just focus on these 2 clustered regions in chromosome 5, we can also see fluctuations in the coverage and copy number. This is an example from Piper, chromosome, like large rearrangement in chromosome 11, leading to a deletion of CDK and 2B, which is a known tumor suppressor gene, and also large insertions from chromosome 14, which leads to fluctuations in coverage. All the examples that I showed you, this both Piper and Oliver, are clonal events. That means they are shared across all the samples that we have sequenced. But we also observed quite high number of private events. That means they're only happening in a single sample. But the largest event that we observed is shared between OT2 and OT6, which we know to be collected in a closer distance in the tissue. We also observed similar pattern in Piper. So this is just a quick glance to this study. For more details, especially for how methylation profile and SNV profile is changing between different samples, please see the poster. I hope I convince you that Severus is the best tool out there. So this is all from me today. So thank you.
And just to quickly wrap up, so we think that long-read sequencing is a really nice technology that can help us to dive deeply into the complicated structural variants in cancer. We developed Severus, which works really well, and it was designed for cancer, for tumor normal and multi-site tumor, different modes. We are also working on a tool called Bahan that I should mention for haplotype-specific copy number calls from long-read data. And we are actively investigating this interesting data from multi-site osteosarcoma sequencing. I wanted to quickly highlight another ongoing project that is driven by our collaborators at Google Health and UC Santa Cruz. They are essentially we collaborating using the same multi-omics cell line panel to train a DeepSomatic tool, which is an extension of DeepVariant for cancer tumor normal analysis and somatic small variant polling in cancer. In the interest of time, I will skip the benchmark, but we essentially are getting very good numbers, and we are now extending this single model to multiple cell lines that we have mentioned previously. And so with that, I wanted to thank you, most importantly, the lab and our collaborators at UC Santa Cruz and Children's Mercy Hospital, and of course, our Nanopore applications team who also contributed to the DOG project. And with that, we'll be happy to take questions if there's time.
Thank you very much for this talk. We've got time for one, maybe two quick questions. Please come to the microphone if you've got a question. I just would like to also mention that we will be implementing that tool in our internal workflow that's going to be on P2Me Labs, and you can also learn more about it at the booth.
Yeah, hi, thank you. It's Mike Demur, very interesting. The question is regarding actual tumor samples in terms of, you need frozen tissue, not paraffin because of the template, but what about tumor purity? You do micro or dissect, and what kind of tumor purity do you need for your sequencing?
For the osteosarcoma, we get a pretty nice purity because it's like an amputation. But for the other cell lines, I think we get below like a 50% tumor purity, and it's working well. But I mean, that kind of depends on what you expect. If you're 50% and low coverage, you may not be able to get enough subclonal events. But for the clonal events, it won't be a problem.
And I wanted to add that you do need frozen tissue currently, fresh frozen. In terms of amount of DNA material, I think we've generated this dog data with 1 microgram of DNA. I think there are possibilities to go a little lower. And also there was an interesting highlight about a couple weeks ago. There was a study led by BC Cancer, and they analyzed a pan cancer cohort, about 200 patients, and all the tumor samples were from needle biopsies. So it's definitely possible to do it even without surgeries and without real biopsies. Of course, assuming you have maybe over 50% tumor purity and it's fresh.
Thank you very much, guys. We will move to the second presentation, which will be given by Julie Geyer. Julie is a postdoctoral researcher at UNC Chapel Hill in the School of Medicine, studying cancer genomics, specifically the genetic underpinnings of pediatric B-cell acute lymphoblastic leukemia. She's part of a multidisciplinary team of academic researchers and clinicians seeking to develop an accessible, reliable, and fast characterization tool for pediatric cancer. Over to you, Julie.
All right. Thank you for being here. My name is Julie Geyer, and I will be discussing some of the work I've been involved in using long-read sequencing to characterize samples from patients with pediatric acute leukemia. So, as many of you probably know, leukemia is cancer of the blood, and it can be subdivided into several lineages, including AML and ALL. Within these lineages, there are lots of different subtypes. These subtypes are really important because they affect patient prognosis and they also inform clinical decision-making. So, for instance, patient A might have a slightly riskier subtype and therefore might require more intense treatment. The work that I'll be talking about today focuses on B-cell acute lymphoblastic leukemia, which is the most common type of acute lymphoblastic leukemia. So the hallmark of lymphoblastic leukemia is the overproduction of these white blood cells. These immature white blood cells essentially crowd out healthy cells, creating the phenotype that we see. We can detect these genomic signatures within these blast cells. And we can detect these signatures at multiple levels. So we can see this at the chromosome level. For instance, we might see different numbers of copies of chromosomes. We can also see this at the gene level. So we often see gene fusions or smaller deletions or duplications. And we understand that the chromosome and gene level at this moment are the clinically relevant scales to be examining these signatures at. So how do clinicians actually determine clinical subtype? They'll do a combination of several different tests, including karyotype analysis as well as FISH. Karyotype analysis is a really standard clinical practice. This helps clinicians visualize the chromosomes and their number of copies. And so if you can see, these blast cells contain 2 extra copies of chromosome 21 based on this karyotype image. Fluorescence in situ hybridization is another clinical assay that is often performed to understand how there might be structural variation at the gene level. So this is a really common way to visualize gene fusions. There are a number of other tests that are really common to detect changes at both the chromosome and gene level. These include things like targeted sequencing, PCR, and microarray. So the primary goal of the work that I've been involved in is to understand if we can use a single assay to get the same amount of information that clinicians are getting from these various multiple tests. So what we did is we did whole genome nanopore sequencing to understand if we could classify these samples. We ran this on about 30 samples from UNC hospitals where there was a known classification. So we know how many chromosome copies are detected, and we also know if there are any gene fusions present. So in order to do this, we start with DNA extraction. So we get a sample from UNC hospitals. We extract the DNA. We then do a ligation-based library preparation. And then we put this library onto a flow cell. One important thing to note is that we employ a technique called adaptive sampling. This is a Nanopore-specific program which allows us to selectively enrich for genes of interest. And so in our case, this is about 60 genes associated with acute leukemia. So in total, these genes make up about 0.3% of the genome. And by using adaptive sampling, we can enrich for these genes about tenfold. And these genes are where we're typically going to see these abnormalities and structural variants. The last aspect of this is to actually run the informatics. So to do this, we'll run base calling, we will align to the human reference genome, and we will then determine or infer digital karyotype as well as detect these fusions. So what do we actually see when we get the data back? So what I'm showing you here is a karyotype plot. And so on the y-axis, I'm showing you the number of reads we're getting. And then on the x-axis, I'm showing you all the different chromosomes. And so we can infer the number of copies of these chromosomes based on the number of reads we're getting back. So we see about 50% more reads for chromosomes 2, chromosomes 4, 5, 6, etc. And so we can infer that there is an extra copy of these chromosomes. Likewise, if you look further over to the right, we see that there's about 100% more reads for chromosome 14, and so we can infer that there are 2 extra copies of this chromosome. So I mentioned that we can look at these changes at both the chromosome level as well as the gene level. And so if we dive a little bit deeper, we want to know if there are changes at the gene level. What I'm showing you here are reads that map to both the BCR and ABL1 genes. So we see these here in blue. I'm showing you just about the first 30 reads that map to both genes. But for this particular sample, we saw about 100 reads that align to both of these genes, which tell us that there's probably a fusion. BCR-ABL1 is a really common fusion with B-cell acute lymphoblastic leukemia. And so this sort of confirmed that this is probably what's going on with the sample. This is another case that we sequenced. We can see here that this karyotype is likely normal. We don't see any relative differences in the number of reads per chromosome. However, if we look at the gene level, we see a number of reads supporting a DUX4 IgH fusion. Again, those are just in blue. The orange and red reads are just wild-type reads. So this tells us that this is likely a heterozygous fusion of IgH DUX4. So in total, we've run about 30 samples. Again, these samples were ones where we knew whether there were gene fusions, and we knew whether there were differences in the number of copies of chromosomes. And the clinically determined karyotype and gene fusion was determined by a combination of usually karyotype analysis, FISH, and possibly microarray and targeted PCR. We saw 100% consistency in what we independently determined to be the classification and what was determined clinically. So this was really exciting to us, and we wanted to move forward and see how this worked in real time. So for those roughly 30 samples, those are what we call retrospective samples. We know the karyotype, we know the fusions. These are ones where we haven't yet determined the classification. So these are active patients, and and the clinical pipeline is still ongoing and was not yet complete by the time we sequenced these samples. So how long does this processing actually take? So the wet lab work, from grabbing the sample, running DNA extraction, and preparing the library, took about 3 hours. Most of that was for the DNA extraction. About an hour of that was for library preparation. I should mention that one of the big differences between our prospective sampling and retrospective sampling is that we're running live base calling and live analysis. And so we're generating reads real time and mapping those to chromosomes and genes. So within about 10 to 15 minutes, we could reliably generate a karyotype. And so from this, we could infer how many copies of chromosomes were in our sample. Detecting fusions took a little bit longer. We could typically detect a fusion in between 3 and 6 hours from the start of sequencing. So in total, the pipeline from getting the sample to classifying the sample took anywhere from 6 to 9 hours. So I want to highlight one of the samples that we did in real time. This is a sample where we can see that there are roughly 50% more reads for chromosome 4, indicating to us that there is likely an extra copy of this chromosome. We also see that for chromosomes 21 and the X chromosome, that there appear to be about 100% more reads for these, indicating to us that there are likely 4 copies of these chromosomes. If we look at the gene level, we see that there are a number of reads supporting a CRLF2 P2RY8 fusion. So we've run about 11 of these real-time samples, and clinically it takes anywhere from about 4 days to a week and a half to determine classification. And so what we did is we would determine our classification, and then the clinic would get back to us and say, this is what we determined. And of those real-time samples, we saw 100% consistency in how we classified the sample and how clinicians classified the sample using things like FISH and karyotype analysis. So we want to highlight a few of the reasons that we're really excited about this. So long-read nanopore sequencing offers fairly low capital costs, and this is really exciting. Clinically, it is quite expensive and laborious to run these multiple different assays. Another thing that's really exciting to us is the rapid turnaround. So as I mentioned, clinically, these assays tend to take several days, up to 1 to 2 weeks. And so we could get a classification in roughly 6 to 9 hours that was consistent with what was being determined clinically. Another advantage of long-read sequencing is the ability to detect structural variation. We're getting longer reads, including repetitive and variable regions, which leads to better structural variation detection. Another aspect of this is we are generating far more data than is necessary for a clinical classification. And we're excited about mining that data for new information. So although we are very excited about the work we've already done, there are some things that we would like to do in the future. So as I showed, right now, a major output is that karyotype graph. And so we can infer karyotype based on that, but we would like to automate this process so that a karyotype in cytogenetic nomenclature is automatically outputted with these graphs. Another aspect of this is we want to understand the limits of detection. We can generate a karyotype very quickly. It takes a bit longer to detect fusions, but we want to understand how can we maximize our resources? How many samples can we run on one flow cell? how many reads do we need to accurately detect these structural variants? Another aspect of this is that this pipeline automatically collects methylation data, which is something that is often used to detect or determine clinical subtypes as well. And so with that, I would like to thank the team that I work with. It is a very interdisciplinary team, and I am one of many. This includes computational biologists as well as clinical researchers, both from the patient angle as well as the lab angle. So thank you very much.